How do you feel about the V8 makes energy drinks? They're like 80. They have like 80 milligrams caffeine in it. It's less than a cup of coffee.
But it's not the caffeine.
They're not carbonated. They're juice.
No, I'm not talking about V8s. I'm talking about regular energy drinks.
Oh,
It's
okay,
not the caffeine. It's all the other products.
You don't have to worry about the other stuff. There's caffeine.
I don't think it cancels out.
Yes, it does. Can we do a unit about the effect of an energy drink on your body?
Maybe. All right, guys, let's go ahead and get started and pick up with photosynthesis, which we introduced last time, and we should be able to finish it today, I think. And then we'll move on to other. Move on to other things. So to review for a couple minutes, and then we'll hit some new stuff, and then I have a sheet for you to work out, which I think will help you for the exam. I think our exam is in 2 weeks, I believe. Is that seem correct? Okay. So it means we have today, we have Tuesday, we have Thursday, we have Tuesday, and then exam. So we'll have today, Tuesday, Thursday, Tuesday. Yeah, so 4 lectures and then the exam. All right. So to remind you, photosynthesis is broken into 2 major components. The first is the light reactions, and the second component is the Calvin cycle. And the main difference between them, well, everything about them is different, but the main difference is that one of them requires light and one of them can happen with or without light. And so the light reactions require light because that's where the energy is harvested by the pigments. And so if it's harvested by the pigments, it has to be where the pigments are, which is the thylakoids, okay? And the thylakoids, when they harvest that light energy, they're going to remove electrons from water. So we'll look at that reaction today. And that releases oxygen, which is, of course, one of the major benefits that we get from plants. And that energy is going to eventually be used to make ATP and NADPH, 2 molecules that we're familiar with from aerobic respiration, although NADPH is a slightly modified version of NADH, but also an electron carrier. The second major component then is the Calvin cycle. And one of the things that happens in the Calvin cycle, which is especially noteworthy, is the carbon fixation. And so converting this CO2 into an organic molecule like a sugar, that is what is meant by carbon fixation, taking that out of the atmosphere as a gas and converting it into a solid that can be used by organisms for energy. Okay, so we also talked about the concept of certain wavelengths of light being absorbed differently by pigments. And when that light is absorbed, one of the events that is going to, what's the right word, color how we feel about photosynthesis is the change in electron energy. So again, this is a weird concept for me to wrap my head around, but hopefully you can believe it yourself, which is that electrons can exist at different energy levels. So you can have a high energy electron and a low energy electron, and the difference between those two can be, difference between those two can be released as heat, can be released as light, or it can be used to do work. And if we absorb electron energy, if our electrons absorb energy from the sun, we cannot do work with that. So that electron energy is released as heat. But if a plant absorbs electron energy and its electrons get photoexcited, it can utilize that energy. Okay, before the electron returns to its ground state, it can harvest that energy to do work. That's the difference between them and us. One of the differences. Okay, and then project that you are working on in lab this week. Did anyone produce an absorption spectrum that looked like this in their lab? Did you all already do the graph? I know I saw some pigments. You didn't achieve that, or you didn't?
We haven't done this one yet. We did like peas and their.
But have you had lab this week? Okay, didn't someone have lab? Yeah, did you all make a graph? Did it work?
Yeah, I haven't graphed it yet. We do a graph inside class.
Okay. Well, hopefully it'll turn out this way.
I don't think it will. I don't think that data's going to look like that.
It always did when I did that lab. I was always really pleased with the
look
result.
like that.
Well, anyway, in the lab, what you're going to do is separate pigment molecules depending on their polar qualities, and you're going to measure their separate absorption. And what we do know, if it does work, or whether the lab works or not, what we do know is that the chlorophylls have 2 spectral peaks where they absorb light most strongly. One is in the blue and green range, and one is in, the blue and violet range, and one is in the yellow-red range. And so because they absorb those colors, whatever is not absorbed is what we see, which is in the green and a little bit into the yellow range. And then beta carotene absorbs in the violet, blue, green, but not in the yellow, red. So beta carotene releases the orange color that we see because it doesn't get absorbed. And that can be translated to photosynthesis. So the absorption means that the pigments are absorbing that energy, their electrons are getting photoexcited, those electrons are being used to do work, which in turn generates energy, which in turn generates sugars and releases oxygen, and so that can be measured. And the rate of photosynthesis, you won't be able to measure this in your lab because they don't have the type of equipment to measure O2 and CO2 consumption. But the rate of photosynthesis corresponds to the places where absorption is highest, right? Because that's where the light energy is absorbed and work can get done. So they should be overlapping, peaks, but what you'll be able to generate, hopefully, is this top graph, and then what will be implied is this bottom graph. Okay, which brings us, I think, where we stopped last time, which is the movement of electrons. And so I'm gonna give you a sheet. I'll just start passing it now, and then when we get to it, actually, maybe we'll start at the back and then we'll end up back. Alone on your island back there. All right, so grab one of those and then we'll work on that here in a second. Bless you. All right, so the movement of electrons should remind you a lot of what you saw when you did aerobic respiration, and the movement of electrons with electron carriers through the membranes of the mitochondria, generating a proton gradient. Okay, and so most of the same things are happening. The difference is where the electrons come from and how they're energized. And so we're going to look today in detail at photosystem 2 and the movement of these electrons. And photosystem 2, as we'll see in a second, is a complex of proteins. And the reason photosystem 2 occurs before photosystem 1 in our linear movement of electrons, well, actually, why do you think that's named photosystem 2 when it occurs first in our process? I'll throw that question out. Audience. Any ideas? This is actually a really common thing with the name of scientific molecules, that the numbers don't seem to correspond with where they occur in a pathway. Any guesses?
Are they discovered then?
What?
Are they discovered then?
Yeah, it actually is named by the discovery. So when we talk about DNA replication in bacteria, polymerase 3 is the main polymerase we talk about, and it was the third one discovered. So instead of calling the main one polymerase 1, we call it polymerase 3 because it was the third one that was found. Okay, so in any case, photosystem 2 is where the electrons initially absorb that light energy, and so there are pigments in that photosystem complex. Thank you. And then that electron is going to follow the journey of this red line, and that's what we're going to diagram on our sheet there in a second. And as it follows that journey, it is going to do work. Thank you. And the main work that's going to be done is the pumping of hydrogen ions into the lumen of the thylakoid. Remember, the lumen is the inside of a membrane-bound structure within an organelle. Okay, and so if we create a gradient of hydrogen ions inside here, they're going to look for a way to go back down their concentration gradient, and that is going to go through the ATP synthase, which is a molecule or an enzyme complex very, very similar to the ATP synthase found in mitochondria, and is doing the same work. Okay, so actually let's go ahead and take a look at our diagram here. So this is one of those things that I hope will be useful to you, like the respiration I hope was useful to you, respiration worksheet. And let's fill it out and pay attention to the way that it's laid out, because like the other sheet, it's not just random boxes. There's a reason it's arranged this way. Okay, so in the the organization of my diagram, photosystem 2 is everything to the left of this dotted line. Photosystem 1 is on the right side of this dotted line. And then we have the stroma, so this is the membrane of the thylakoid going out into the stroma. And remember, the stroma is the outside of the thylakoid, right? So in this case, everything that's brown is the stroma. Everything that we're looking at inside this is in the thylakoid. Okay, and then in the middle here, we have the lumen, which is the inside of the thylakoid. All right, and if you notice too, this is set up kind of like, somewhat like a graph, which is that as energy increases, molecules are higher up on the page. So these molecules have high energy, and these molecules have lower energy, okay? All right, so if we start from the left side, the first thing that happens is an event called water splitting. And that is because water is literally being split into hydrogen ions and oxygen. And when water is split, an electron is removed, and that electron is donated to a molecule called P680, right? And P680 is named P680 because it is a pigment, that's the P. And the reason it's named 680 is that 680 is the wavelength of light that it optimally absorbs. Okay, so what happens when P680 receives its electron? Well, the first thing that happens is it gets energized by light from the sun. And when it is energized, it goes by a different name. And so we call the energized photoexcited version of P680 P680 star. It's just a little asterisk, but that indicates that it's the same molecule, but in a different energy state, which in this case is photoexcited. Okay, P680 star with its high energy electron is going to give that electron away. So each step in this diagram is the movement of an electron from one thing to another. So the first thing that it's going to give its electron to is a molecule called, that's right, PP, which stands for pheophytin. It's a strange name, pheophytin. Okay, and pheophytin is an electron carrier. I'm going to put carriers because we're going to have more than one. All right, pheophytin is going to transfer this high energy electron to another electron carrier called QA. Q is an abbreviation for quinone. I think I can spell that. Right? That's right. And QA is going to pass that electron to a second quinone carrier, which is called QB. Okay, now QB, as you notice, is right at the boundary of our photosystem in my little diagram. And that means that QB is going to be taking this electron out of the photosystem and donating it to a structure called the cytochrome complex. All right, the cytochrome complex, you want to make some notes on your sheet there. The cytochrome complex exists in mitochondria and in thylakoids. Oh, yes.
We don't have enough boxes.
We only have 3 boxes instead
We
of
only have-
4.
What? Yeah. Which box did I miss?
We only have 3 boxes.
Oh, shoot, I printed out an old copy. Damn, it's too early in the lecture to make a large mistake. Okay, well, I have 4, no I don't, I drew it in on mine. Shoot, that explains why I drew it in. Okay, I have a suggestion. Draw in a box. This is your art challenge for the morning. Thank you for drawing that to my attention. I will see if I can find the right version. I guess I could print it out straight from the PowerPoint. Lo siento. All right, are we back on board? We've got our QB. Okay, we've overcome this challenge. Good work, team. My apologies. Okay, so once we get to the cytochrome complex, what I was going to say is that the cytochrome complex is a type of protein complex that we saw in the mitochondria and we're seeing again in plants. So they are actually very closely related, even though one is plant and one is animal. But what the cytochrome complexes do is they use energy from the electron to pump hydrogens. So they are one of our proton pumps. Let me see if I can say that succinctly. Uses energy from electron to pump hydrogen ions, which I will also call protons, into the lumen. Okay, so this is one way that we are creating an electrochemical gradient. They're actually going to end up being three ways, but this is one of them. Now, as the cytochrome complex uses that energy, does that one have this? Have I modified it? I must have pulled this out of an old stack. Now the rest looks okay. Okay, well, shoot. Oh well. Life rolls on. So we're creating that electrochemical gradient. Now, meanwhile, our electron does not get used up, right? It doesn't become part of the proton. It can't become part of the proton. That would not be a proton anymore. Instead, that electron is going to be handed off by the cytochrome complex. You can see its energy is waning. And so it leaves the cytochrome complex in an electron carrier called PC. Up here so you can see it easier. And PC stands for plastocyanin. Okay, so it's another electron carrier. Remember what electron carriers do. They're unusually good at taking an electron from one molecule and passing it off to another. And this is a minor, well, it's not a minor side point, but it's one I won't ask you about, which you'll probably encounter when you take, or if you take chemistry, or you may be taking chemistry now. The concept of electronegativity, we talked about how some atoms are more electronegative than others, and they tend to draw electrons to them and share unequally in covalent bonds. One of those concepts of electronegativity plays out in the electron carriers, which is that each electron carrier is a little more electronegative than the one before it. So it's pulling that electron towards itself, and that's why they get passed along like a hot potato. Anyway, just a little aside. So our plastocyanin now takes the electron from the cytochrome complex, and all this is happening in the membrane of the thylakoid. So we'll look at the picture again to reaffirm the stuff. So what's going to happen to this electron now? It's going to reach its low point, right, energy-wise, and when it reaches that point, it's given away to a molecule called P700. All right, I guess while we're labeling things, let's put another category here, which is pigment molecules. And so our two pigment molecules in this process are P680 and P700. All right, so we know what pigment molecules can do. They can absorb light. And we know what photoexcited electrons do. They can increase their energy levels. So that's what happens. Our P700, holding those low energy electrons, receives energy from light, channels that energy in a way we'll talk about in a second to the electron, and now that electron energy level goes up. And so our P700 with its now high energy electrons rises to its place on our diagram, and now it's going to give those electrons away to do some work. In order to give those electrons away, it needs to pass it to, you guessed it, an electron carrier. This electron carrier is called ferredoxin. I need to look up the origins of these names. I've never done that. Pretty interesting names. In any case, ferredoxin, our electron carrier, takes the now high energy electrons from P700 and gives them to the last enzyme in our it's called a Z scheme. I'll explain that in a second. Gives it to the last enzyme, which is NADP plus reductase. And we mentioned this one last time. The name NADP plus for this is an electron carrier that does not yet have its high energy electrons. And if an enzyme is a reductase, to reduce something means to give electrons to it. So NADP plus reductase means this enzyme gives electrons to NADP plus. And by so doing, that creates the molecule NADPH. And actually, that's a little misleading. It's not NADPH plus, it's NADPH plus hydrogen ions. Does that make sense? That's not a superscript. I didn't realize that was misleading until I saw it on the screen. So what it's actually saying would have been better if I had done it this way. So those two things. So the process of producing NADPH results in a hydrogen ion. And there's a reason I'm mentioning that, which I'll show you in just a second. Okay, so this process was discovered surprisingly long time ago. I think it was in the 1940s or 1950s. And how people did such ingenious research with so little technology just speaks to the intelligence of those scientists. But it was named the Z scheme, and the reason it was called the Z scheme, it's Z as in the letter Z, is because the energy level fluctuates from low to high to low to high. So it looks like a Z. So if you were to graph this on a chart based on its energy, or based on the energy of the electron at different stages, it would look like a Z. So they call it the Z scheme. Okay, let's look back. Hold on one second. Let's look at this again and now compare. Now that you have this in your hands, let's take a look at how this might appear in a more three-dimensional diagram. And so our photosystem 2, right on the left side of our chart, here's water being split by photosystem 2, so-called water splitting enzyme. Electrons are being given away to P680. P680 absorbs light energy, then passes that electron on to our three carriers, pheophytin, QA, QB, the mysterious boxless QB. QB leaves photosystem 2, and as it travels through the membrane, so all these things are membrane associated, if you notice. Everything is attached to the membrane, okay? As it moves through the membrane, it gives it to cytochrome complex. The cytochrome complex is a transmembrane protein, so it's able to move something from one side of the membrane to the other, and the thing it's moving is the hydrogen ions, okay? So it is a proton pump. It gives this electron to another electron carrier, plastocyanin. Plastocyanin gives it its electron to P700, boosts the energy from the light, and then P700 passes it on to ferredoxin. Ferredoxin is going to take it to the last enzyme in this system, which is NADP plus reductase. And NADP plus reductase reduces NADP plus. So it adds an electron to that, actually two electrons, and then two hydrogens, which are reactants in this chemical reaction. One hydrogen is incorporated into the NADPH, one hydrogen is released. Okay, and so the number of hydrogens, if you notice, goes down on the outside of the thylakoid. That's important, all right? Meanwhile, our hydrogen ions, which are increasing in concentration in the lumen, have created an electrochemical gradient, as well as a change in pH. If you think about what's happening in this lumen, not only is a gradient being made, but remember, pH is completely dependent on hydrogen ion concentration. So some people, I wouldn't worry about writing this down, but just, if you see it maybe later in another class, some people talk about what's happening in the lumen in terms of delta pH, which is the change in the hydrogen ion concentration, which is also a chemical gradient. In any case, then those hydrogen ions leave via the ATP synthase, and we make energy. Okay, so the major outputs of this, as diagrammed up here, are oxygen, which came here, ATP, which was there, and the NADPH, which was there. Those are the things. Well, the oxygen is going to be used by the plant to do aerobic respiration, and it's going to be used by us if it makes it to the atmosphere. And then the ATP and NADPH are going to be used elsewhere in the chloroplast for the Calvin cycle. How do we feel about that? There's a lot happening. You could do this though, right? You've demonstrated you can do that. So now I know what you're capable of. And you're definitely capable of that. Okay, let's ask a question. There's the little section in your book. If you read if you read this chapter, which I know not everyone buys into the reading books thing, it's true. It is true. Not everyone reads anymore. It's too easy to do things without reading. But this is actually a good chapter in terms of relating what's happening in a pretty, it's very clear and direct, and it doesn't go off on a lot of deep chemical tangents. I was looking over it this morning, and I just was thinking that of all the chapters about photosynthesis, this one is actually really good. So I would encourage you to read it and fill in the dots, or fill in the blanks maybe that you may not be getting in lecture or feel like you're understanding right now, because it'll maybe sew everything together. That being said, the last couple, maybe 3 or 4 pages of the chapter are about unusual plant events, not unusual, but different than the traditional photosynthesis. There's C4, I think it's called, and plants that use the CAM system. I'm not going to go into that stuff, so if you start wading into the last page of the chapter and it looks really different than what we're talking about, read it if you want, but understand you're not going to be responsible for that, only what you see in class. Okay, but in the book, the reason I went on that little bit was in the book they show this diagram and they talk about this process of cyclic phosphorylation. It's actually worth thinking about, and so I put a slide in there about it. And cyclic phosphorylation is a contrast to the linear movement of the electron that we see here, right? So that's a linear electron flow, but in cyclic phosphorylation, the electron is just doing loops in the thylakoid membrane. So what is happening? Well, one thing that is produced in this photosynthetic process, in the light reactions, the first half of photosynthesis, is ATP and NADPH. Okay, but it's possible that the cell will have enough ATP and not enough NADPH, or it will have enough NADPH and not enough ATP. So it looks like everything, if we just keep on making this as it's written, we'll just have everything we need and it flows smoothly into the Calvin cycle, but the amounts that the cell needs of these two things won't always be the same. And so in that case, sometimes the plant will employ a little trick to increase the amount of ATP it produces if it already has enough NADPH. Okay, so if NADPH is not an issue for the plant, if it's got what it needs, it will channel its resources strictly into making ATP for a little while. So this can't continue indefinitely, but it can sometimes occur to fill in the lack of ATP. And so it's actually a pretty simple concept. If we can understand the other one, this one makes is much easier. What happens in P700, normally, P700 is going to give its electron to ferredoxin, which gives it to NADP plus reductase. But in cyclic phosphorylation, the ferredoxin, instead of giving its electron away, takes its electron back to an earlier stage, gives it to QB, and remember, it's just been juiced up by this light, so it's got some energy. So that electron goes to the cytochrome complex, which pumps some protons, and then passes on to plastocyanin, then it can keep doing this, or ferredoxin can take it on, down to its permanent destination. And so it's kind of a little sideshow going on in the midst of the other linear electron flow when you need more ATP. So it's called cyclic phosphorylation, which is a pretty descriptive name because of course it's cycling, and phosphorylation refers to the production of ATP, right? You add a phosphate. And so that's what's going on there. Okay. Now, one part of this that we want to look at in a little more detail before we hit the Calvin cycle is the photosystems. Okay, and specifically photosystem 2, which again is the first one in the linear movement of that electron. And so photosystem 2 looks startlingly like the head of a gigantic ant looking at us, but this is a complex of many different proteins and many different pigments. So all these little squiggly guys represent pigments. Remember how the pigment had structure at the top, which was, what was it called? The phytol head? I won't sit back, I'll just flash to this, straight to that slide. Oh, the phytol is the tail. So it has this structure at the top called the porphyrin ring, and then the phytol tail. Okay, and that's what is diagrammed in the giant ant face. And remember, this thing that we're looking at right now is this. Okay, I know there's a lot going on. All right, so what is happening in that photosystem? Well, it's a cluster of pigments, and not all of those pigments are equal. Okay, some of those pigment molecules have a special role that the others don't have. And so collectively, this is called the light harvesting complex. Remember, this is in the thylakoid membrane. If we could see the membrane, it would span across like that. And when light hits this membrane, the pigments will absorb it, right? That's what the pigments do. By definition, they absorb light. And so when they absorb light, the electrons in those pigment molecules that have absorbed the light will go to a high energy level. All right? Now, the thing that happens next is kind of wild. Instead of passing an electron from one pigment molecule to another, right? That's one possible thing you can do, but that electron, or let me rephrase it. All of those pigment molecules have electrons that are capable of moving to high energy. So instead of giving an electron to a pigment molecule that already has an electron, instead it just raises the other pigment molecule's electron energy through this thing called resonance energy transfer. Okay, so if it gets close enough to another pigment molecule, the energy from one of those high energy electrons can charge up another electron and make it high energy. And when that happens, energy leaves one pigment and goes to another. All right, so they don't all get charged up. It's a little misleading in the diagram. They're not all energizing. Instead, this energy is being passed from one to the next. Okay, and then eventually, that that energy in the high energy electron of the electron energy transfer is going to reach a very special pigment molecule, which we met earlier, which was P680. Okay, and when P680 takes on that high energy from the resonance transfer, its electron goes to a high energy level and it takes on a different name. All right, so P680 and P680 star are both the same molecule, but they differ in their energy level. So when P680 star takes on this high energy electron, it becomes very unstable. It needs to get rid of that energy. Now, if it doesn't get rid of the energy will just dissipate. Just like when we absorb energy from light, it just dissipates, right? We lose it. And so that's possible. But what the plant can do, and usually does, is capture that energy before it's lost. And so to capture that energy, it's going to transfer it to something called a primary electron acceptor. And for reasons that are not clear to me, they don't label this in that diagram, but the primary electron acceptor is pheophytin. I'm not sure why they don't just call it that. But in any case, the primary electron acceptor can hold on to that electron energy in a way that the pigment molecule P680 cannot. And so once we get the electron there, we're stable. Okay? And so P680 then, as you probably recall, is going to transfer that electron to QA, then to QB, and then it will leave the photosystem. Okay, so once we get it to the pheophytin, we're good. All right, now we do have an issue left to deal with, which is that our P680, once it gives away an electron, it becomes P680 plus. Okay, there's a part on our sheet we'll fill in about this in just a second. And it becomes P680 plus because it's lost an electron, right? So if you lose an electron, that's a negative charge that you're losing, which means your net overall charge will be one more positive than it was before. So once it becomes P680 plus, it's no longer P680 star because its energy has come down. Bless you. Once it becomes P680 plus, it needs to return to its original state so that it can function again and absorb that high energy from the resonance transfer. So in order to become what it once was, it needs an electron, right? It gave its electron away, it needs to get one back. And so where's it gonna get that electron? Water. It's the perfect solution. And so water, when it's split, breaks into oxygen gas and hydrogen ions. And in that process, 2 electrons are stripped away and given to P680. Okay? And so when those electrons return to P680, our electron returns to P680. Now P680 is back where it started. Okay, it's low energy and it's ready to receive that resonance transfer. So P680 is going through a little cycle of its own. Okay, so on the back of your sheet, hopefully this is copied correctly, on the upper left side, there's a little diagram that tracks P680's movement through these 3 stages. Remember, this is all happening in photosystem 2. So the changes that happen in P680, the first thing that happens is that light energy charges P680's electron into a high energy state. So it becomes P680 star. All right, P680 star, remember, is a very unstable molecule and is looking for a place to give its electron away to. And the thing it's going to give its electron away to, remember, is the primary electron acceptor, pheophytin. And so that electron is transferred to pheophytin, which takes P680 and converts it to P680 plus. P680 plus needs to return to its original state. And so photosystem 2 is going to use an enzymatic component of that large protein complex to split water. And when it splits water, water will release oxygen gas. And remember, one water molecule will only release one oxygen, but oxygen never exists as just one oxygen atom. Or it doesn't exist freely as one oxygen atom. And so that's why they have half of O2, because oxygen will only ever exist as O2 when it's by itself. In any case, half of O2 and then 2 hydrogen ions. And then we're back where we started. And so the electron from the water goes back to P680, and then we're back in business. Makes perfect sense, right? Would you call this process common sense? Not going to just wander in and figure it out. It's crazy that scientists have been able to work all these details out. It fascinates me so much. This is what the photosystem 2 actually looks like if we diagram it, not we, but if scientists diagram it using this common, what would you call it? I think people call it ribbon model of proteins. So all of these little guys are alpha helices. If you remember that term from earlier when we talked about protein structure. I've got a few beta sheets in there. There's another beta sheet. And so these alpha helices are going to be existing in the phospholipid bilayer of the thylakoid membrane. But lots of stuff going on there. Very cool. Okay, and this is, just to reiterate, the Z scheme. So this is a different version of the diagram that I gave you, but showing the same thing. P680 getting its, oh, it is 2 electrons. I thought it was 2 electrons. 2 electrons from water. P680 gives this electron to pheophytin, not labeled here, but that's the primary electron acceptor. Gives it to QA, to QB. The cytochrome complex uses that electron to pump hydrogen ions across the membrane, then gives it away to plastocyanin, which gives it to P700. Same thing happens in photosystem 1 that happened in photosystem 2 with the resonance transfer. P700 is the lucky recipient of the final energy blast. Then that gives it to an electron acceptor, which gives it to ferredoxin, gives it to NADP plus reductase, and we make our NADPH. Okay? All right, before we do the Calvin cycle, there's one part of this, the top right of the same page where you just wrote the P680, which is why does the proton concentration increase in the lumen and decrease in the stroma? Hold on, I was looking at the exam inventory is already posted, by the way. Somewhere in here, I thought I asked specifically this question. Well, it just says, be sure you know the worksheet. Okay. And it says, and had as a quiz, because last spring I did have it as a quiz, but I don't think we're going to do a quiz on that one, because we only have one quiz to go. Can you believe that? I really misscheduled that. Oh well, is what it is. Okay, but in any case, the question says, why does proton concentration increase in the lumen and decrease in the stroma? Let me put this back up and we'll look at it. So there are three events that are happening, and I'll write them up as we go along. Okay, so the first event that happens that increases hydrogen ions in the thylakoid versus the stroma is the splitting of water. Okay, so say it this way: water splitting releases hydrogen ions into the lumen. Okay, the second event is going to be the role of the cytochrome complex, right? Once that high energy electron moves through the cytochrome complex, energy is pulled from that electron to do the work of pumping hydrogen ions against their gradient into the lumen. So step 2 would be the cytochrome complex pumps hydrogen ions into the lumen. And the third one is a little more subtle, but is important. So both of those two things are fairly active ways to increase the hydrogen ion concentration in the lumen. You're pumping it in and you're releasing the ions from water. The third way is what's happening over here in the right field. We have the formation of NADPH, and this chemical reaction involves 2 hydrogen ions in the reactants, but in the products only has 1. Okay, so there's a net loss of hydrogen ions when that electron carrier takes on the electron. It will also take on a hydrogen ion. And so the third thing, which I would put as our concentration changing, is the formation of NADPH removes hydrogen ions from the stroma. Okay, so remember, there are 2 ways you can change a gradient. You can add things in one spot, or you can remove them from another. Either way, you're tipping the scales and changing that balance. Okay? All right, so that's to make sure I'm not forgetting something on there. Okay. All right, now let's turn our attention to the second part of photosynthesis, and this is not quite as involved. If you're thinking that we're only halfway through, we're not. We're much more than halfway through photosynthesis. So the second part is the Calvin cycle. And the Calvin cycle is called a cycle because, like the citric acid cycle, one of the components that you start with gets remade in the process so that this chemical reaction continues in a cyclical pattern. And the thing that gets remade is this 5-carbon molecule called ribulose bisphosphate. Okay, and most of these we're going to go by the abbreviations, but ribulose bisphosphate gets remade, and that becomes the first starting material for the Calvin cycle. Trace over here. The Calvin cycle is split into 3 different parts, and remember the purpose. Don't get lost in the details here, even though the details are important. The purpose of the Calvin cycle is to fix carbon, right, which means carbon dioxide will get incorporated from being a gas into now a solid. So that's purpose number 1. Purpose number 2 is to produce sugar precursors. So this says glucose and other sugars, but if you notice, that's later, right? That's not part of the actual Calvin cycle. The Calvin cycle produces this molecule called glyceraldehyde 3-phosphate. That's a sugar precursor. Those are like the building blocks to make glucose, fructose, starch, cellulose. And so it's not just making glucose, even though we often simplify and say that it is. So those are the 2 main goals. Get rid of the CO2 and produce sugar precursors. And if you notice, this is going to take a ton of energy. So 12 ATP here, 6 ATP there. We're going to use up all the high energy electrons from 12 NADPHs. So there's a lot going into this. But that's okay, because we have the sun to power this process. So we can use a lot of energy. All right, so let's split it into 3 things. Let's see how I have it set up on here. Reaction. All right, let's fill in our page as we go along. That'll make it a little easier than circling back. So in the Calvin cycle, if we want to start out with the full reaction, and when I put the reaction on here, I'm not so interested that you can balance this and have all the numbers correct. I'm interested in you knowing what goes in and what comes out. If you want to know the numbers, that's great, that's awesome. And I'll put them up here. But mainly I'm interested in what goes in and what comes out. So we know carbon dioxide goes in. We use ATP at 2 different places. So there's a 12 and a 6 ATP investment. We use a large number of NADPHs, 12 to be exact. And then our starting material is ribulose bisphosphate. Okay, so all of that is in the reactant side of our chemical reaction, so it's a lot. The result, I'll put it on the second line. We have glyceraldehyde 3-phosphate, and we actually have 2 of those. I'm going to put a 2X there to the left. And we have the product of using up all of the energy molecules we have. So 12 NADP pluses came off, right? Because we used the high energy electrons from those. We have 18 ADP. So we used those ATPs for energy, and they were hydrolyzed. And then in addition, and this is part of why it's a cycle, we end up with RUBP. So RUBP goes through a lot of changes, but in the end, we remake it to get the cycle back up and going. Okay, let me diagram the way you've got it on your page there. So we have our inputs, we have our energy, and we have our outputs. And then we have phase 1, phase 2, okay, this is going to be less complicated than you think when it's all said and done, I promise. It's just going to look complicated at first. All right, so what goes in phase 1? So phase 1 is the carbon fixation, right? Maybe we should write that to the left here. So this is the name, bless you, of the phase. So phase 1 carbon fixation. The RUBP is our input, right? That's what we start and end with. And since phase 1 is carbon fixation, we can't remember our carbon dioxide. Now there's no energy used in the first phase. Okay, so this is actually a exergonic reaction. And the enzyme that catalyzes this is something you'll want to remember, is Rubisco. This is the only enzyme in this whole process that I'm going to ask you to remember. And Rubisco has an impressive full name, which is why people call it by its nickname Rubisco. Its full name is ribulose bisphosphate carboxylase oxygenase. And Rubisco has the claim to fame that it is thought to be the most abundant protein on Earth, which is quite a title. Because it's involved in photosynthesis in photosynthetic bacteria and photosynthetic trees, and anything green out there, aquatic or otherwise, has this enzyme. And so it's everywhere. And another reason why there's so much Rubisco in the biosphere is that Rubisco is apparently a slow enzyme. And so because it's such a slow enzyme, plants have a lot of it because it's not fast enough to just have a few Rubisco molecules in the chloroplast. So because of its slow speed and because of its ubiquitous spreading out in all of the biosphere, it is thought to be the most common enzyme. Okay, so we put in RUBP, we put in CO2, there's no energy involved, and so our output is going to be 3PG, 3-phosphoglycerate. And we're just going to use abbreviations. That's enough. Okay, the second one, a lot happening here, but we can wade through it. So in the second one, our input is going to be the output from the previous one, right? So we outputted phosphoglycerate, which is what's going to be the input for this. And, oops, I put that under energy. Sorry. Should be over there. So our 3-phosphoglycerate is going to go into phase 2. And additionally in phase 2 we have 12 NADPH, which are going to contribute their electrons. And this is the most energy intensive step in the process. So of the 18 ATP we use, 12 of them are going to be part of this event. So we'll put something in our energy category there, which is 12 ATP. All right, the product of this is really important because this is the precursor to sugar production. And so the product is glyceraldehyde 3-phosphate. And I'll put a little star there. Say sugar precursor. I guess a better term would be carbohydrate precursor, but that's okay. We can go with sugar. All right, so we have our glyceraldehyde 3-phosphate, and I'm going to kind of put NADPH, maybe I'm going to put that in parentheses, because it's not part of the organic molecules that we're really tracing. And so, because the NADPH got used up, we end up with NADP plus over here, 12 of them. I'm going to put those in parentheses. Oops. All right, now phase 3's input is going to be the output of phase 2. So the glyceraldehyde 3-phosphate, some of that is going to be used to make carbohydrates, but some of that has to be channeled back into the process. And if you look at the amount, there are 12 of those molecules made, 2 of them leave to become sugar, 10 of them get reused. So it doesn't seem like a very efficient process if you're only going to be using 1/6 of that product to go on, but this is what it is. And so the glyceraldehyde 3-phosphate goes back into phase 3, and the output is going to be the regeneration of ribulose bisphosphate. And this is going to use our other energy input, 6 ATPs. Okay, so it looks like a ton of stuff, but let me show it to you in a different way. So if we're trying to learn that chart, which I hope you will, we can think of it as in terms of phases too. So phase 1 is RUBP and CO2. So what if we write it like this? RUBP plus CO2. And then when we go into phase 2, we have 3-phosphoglycerate, which is the product of phase 1. I'm going to leave the NADPHs out. And 3-phosphoglycerate is going to go into our last phase to become RUBP. Right? So that whole chart really is telling us this. And we could put our NADPHs and ATPs in there if we want. But I'm trying to show you that it's not quite as bad as it may seem. All right, am I leaving anything out? I don't think so. Oh, I am leaving one out. I left out glyceraldehyde 3-phosphate. Okay, I'm falling apart, it seems. And that gets us back to RUBP. There we go. RUBP. ATP, CO2, the phosphoglycerate, glycerolic phosphate, RUBP, and we're back where we started. Okay, so not too bad, I hope, or I hope you don't feel that that's too bad. And if we look at this in the scheme of where we started, if we sort of wrap things up and connect the dots, remember our light reactions produce the ATP and NADPH, which are needed, right, for the Calvin cycle. It also takes into account the carbon dioxide, which is in the atmosphere, both because we are exhaling carbon dioxide. Plants are also producing their share of carbon dioxide. It's just that they produce more oxygen in excess. And then what else? And then when it's done, the NADP, which is generated in the Calvin cycle because they donate the electrons, and the ADP, which is made because of the ATP hydrolysis, that can then go back to the light reactions to be regenerated. So all this stuff is flowing back and forth. Okay, and we already did that, and we are now into the blackness of my soul. Who wants to start a new topic?